This presentation describes Chapter 8 of the new ASE Teaching Secondary Science Biology book. The chapter is called Variation and was written by Paul Davies and myself, Neil Ingram. Paul cannot be with us today, but I will walk through and talk about our chapter. The chapter is built around the learning progression outlined by the national curriculum for Key Stages 3 and 4, building on the work of Key Stage 2, especially in Year 6. There, the emphasis is upon variation, that living organisms have lived on this planet for a long time because they show variation. Variation means they show differences between themselves and between themselves and their parents. These differences influence their ability to survive in particular environments. This is significantly developed at Key Stage 3, where the ideas of DNA and chromosomes are introduced. Key Stage 4 develops the idea of Mendelian inheritance, polygenic inheritance, and also ideas about the genome. What we're hoping to do is to make more of the experiences of looking at the cell nucleus at Key Stage 3. And rather than simply saying that's the nucleus, explain why that nucleus is such an important part of the cell. What we're aiming to do in the whole chapter at both Key Stages 3 and 4 is summarised by this quotation from the chapter, where Paul has written that we are helping students to make the link between the visible structure and functions of the nucleus and DNA and the invisible abstract ideas of genes, development and natural selection. We are introducing ideas which will be taken up in later chapters of the book, and there are considerable cross-links in the book between different chapters. A second major potential barrier to learning is the complexity of the language that's used in genetics, and this is something that we explicitly address in the chapter. One of the problems that students and indeed teachers have when thinking about the nucleus is the different scales on which the components exist. We range from tiny DNA nucleotide bases through to DNA molecules and chromosomes and to the genome, all inside the nucleus. And yet the genome only occupies less than 1% of the volume of the nucleus. And alongside that are numerous other proteins and nucleic acids which help the cell to use the information in the genome. So the nucleus is a busy and an active place. The chapters have certain key features which we hope will be useful to teachers, such as regularly considering prior knowledge and experience, and teaching sequences. There are regular boxes distributed throughout the text. This one is about scientific literacy. There are key activities which can be used directly in the classroom. There are links to science in a wider context and career opportunities. Links to mathematics, which is particularly important in the area of genetics and genomics. We have expanded our thinking on genomics to include some of the latest thinking in the subject, which will prepare teachers and students for a world where genomic testing is becoming more commonplace and the uses of those tests are impacting upon the way in which citizens can live their lives. Children who were born in the year 2000, when the first genomic studies were published, are all now adults and part of the genomic generation. It matters significantly that we all understand as citizens that only 1% of the genome carries information needed to build all of the proteins in our bodies. It also matters that we understand that all the genetic differences between us in the world amount to less than 1% of the total genome. In other words, most of our genome is the same in all humans. And indeed, much of our genome is the same in other organisms too. Not just the great apes like monkeys and gorillas, but all living creatures. All life is genetically related to a single living organism that lived on Earth about 3.5 billion years ago. And this fundamentally can change our thinking about our place and role in the world. The old misconceptions that the nucleus is the brain of the cell or that DNA is the book of life are being replaced by a new model in which the genome and the cell cytoplasm interact together to create all of the phenotypic characteristics that we possess, some of which differ between us. In this chapter, we're providing background information to enable teachers to explain these new ideas to their classes. We cover more familiar topics too, such as Mendel, single gene inheritance, and sex determination. This is ethically quite sensitive these days, when many people are questioning their biological identity. We have taken the approach that says that a person's biological sex is determined at conception by the arrangement of the sex chromosomes, but a person's gender arises as an interaction of their genome with the social world around them. Gender identity can develop and change throughout a person's life. A walk through the key ideas at Key Stage 3 and Key Stage 4 show how closely they match the national curriculum. The chapter ends with an extensive bibliography of additional resources, both websites and text.